The last class of biomolecules is called nucleic acids. So we have two main types of nucleic acids. Now there are more nucleic acids that we will encounter in later in the semester, but for right now you just need to remember two major types of nucleic acid. So we have DNA and RNA, and make sure you know how to spell out the entire word, like what DNA stands for, right? D stands for deoxy, ribo, and then N stands for nucleic, and then A acid. Okay? And then the only difference between DNA and RNA is this first one, right? So when you look at it, there's an additional deoxy in DNA, and then we'll talk about what it means, okay? And then that's going to help you remember what DNA and RNA stand for. Okay? So the nucleic acids, especially these two, are so important because they carry the genetic blueprint for a cell, right? So those genetic blueprint will act as the instructions for how to build the cells and how the cells function. Okay? We are going to first look at the monomer of nucleic acids. So nucleic acids can be very long chains, so they can be really, really big. And those long chains, those polymers are made up of monomers, right? The subunit. So for DNA and RNA, the subunit is called the nucleotides. Nucleotides. So remember that, this is very important. And this picture over here shows you the typical structure of a nucleotide, which is made up of three components. So you have to know this. On the test, I may ask you to identify each component. Okay? So I may give you this structure, you tell me what kind of biomolecule this is, and your answer will be nucleic acids, and then you're going to try to identify the three components. Okay? So let's go through each one. This is a nitrogenous base. And nitrogenous just means it contains nitrogen, right? So you can also call it nitrogen-containing base. So you can see how many nitrogen atoms are in the molecule: 1, 2, 3, 4, 5. Okay, so this is the base. Now the base is going to be very important when we talk about DNA in more details later this semester. You will learn that the base can change because you have 4 different nitrogenous bases in DNA, and also 4 in RNA, and then when the base changes, that's how you get different DNA sequences. Okay. The middle one is do you remember what this is? It's a sugar, right? It's a pentose, right? 5-carbon sugar. So this is also carbohydrate. So you do have carbohydrate or carbohydrate component in your genetic material. Okay. We're going to look at the difference between DNA and RNA. Oh, you know what? I'm going to save it for next slide. Okay, so this is a pentose, and then the pentose is in the middle, and then on the other end, on the more exterior, when you build a DNA, molecule, this group, the phosphate group, is going to be on the exterior. So phosphate group, remember, it's very polar. Okay, so if it's a polar, I'm going to ask you, what kind of charge does it carry? I'll give you a few seconds. So it should be negative charge, right? You see the negative charge on the oxygen atoms. Okay, so the phosphate group carries negative charge, and the other two components do not carry any charges. So that means usually the DNA molecule is negatively charged. Okay? And then it's going to be polar, so it can dissolve in water pretty easily. Okay? So you're going to use that information when we do gel electrophoresis in the lab. All right, let's move on to the next slide. All right, so this is more details about the structure. So we're going to look at the pentose to differentiate DNA and RNA. So you can look at this number 2 carbon. So this, you see it shows you this number 2, number 3, because you start counting from here, right? Number 1, number 2, number 3. Now the difference between DNA and RNA is in this number 2 carbon. What kind of functional group is attached to number 2? If it's RNA, then it's a hydroxyl group, which contains oxygen. But when you look at DNA, there's no oxygen, right? So just a hydrogen atom. So oxygen is not present, so we call it deoxy. So D means none, no, right? Oxy means oxygen. So that's the difference. Oxygen or DNA doesn't have an oxygen connected to number 2 carbon. Okay, so that's the difference. Now, when you look at the nitrogenous base, here it shows you 5 nitrogenous bases. Now, you only have a 4 in DNA and 4 in RNA, and that's because in DNA you have a thymine, but in RNA, thymine is replaced by uracil. So there's no thymine, but instead you have a uracil. So you have 4 nitrogen space in DNA or RNA. Okay. Now there are 2 classes of nitrogen spaces, 2 groups. The first one is called pyrimidines. So that includes cytosine, which is usually represented by C, and you have thymine T, uracil U. And then purines, there are only 2, adenine and guanine, okay, A and G. Okay, so you need to know which one's purine, which one's a pyrimidine. So this is how I remember it. Now we know that agriculture is very important, right? And then when you say agriculture, when you refer to agriculture, a lot of people may just say or not say it, but use AG to stand for agriculture. So AG, you can kind of group them together. So those are purines, and then the other 3 are pyrimidines. Okay, so when you have all these nucleotides, okay, so this is one nucleotide, right? Now you are going to link them together to form very, very long chains. This is a DNA formed by all those nucleotides. Now you can see that there are 2 chains, right? So this is one chain and this is another. So DNA consists of 2 chains, and those 2 chains will form a double helix structure. And this structure is actually very important. This kind of ensures that DNA can be replicated, right, in a very conservative way. So you try to minimize any alteration, any mutations. You're making exactly the same new DNA as the old DNA. Okay, so we'll talk about all those in details when we get to the DNA chapter. Okay, let me go here and then we'll go back. So when you have 2 strands, right, all linked together, that means certain part of each nucleotide has to kind of pair or linked with the counterpart on the other chain, right? So we're going to use the nitrogenous bases to kind of pair with each other to link the 2 strands together. So here's the rule: A pairs with G, and C pairs with G. So when you come up here, so if this is A, then this has to be a T because A pairs with T. If this is C, then this has to be a G because C pairs with G. Okay, and then between those nitrogen spaces, you are going to have a hydrogen bond, a very weak chemical bond, to link those nitrogen spaces, which also kind of keeps or holds the 2 strands together. Okay, so this is very important. Okay, you absolutely have to remember that. So this is called the base complementary rule. You need to remember which one pairs with what. Okay, now RNA, so ribonucleic acid. It doesn't really directly, store your genetic information like a DNA, right? So to help you use the genetic information, the instructions in DNA to direct the synthesis of proteins. Okay? So when we say gene expression, we are referring the process to the process where you are using the information in the DNA to make proteins. So proteins are, if you remember, performs a wide range of functions, right? So in your body, when you look at the structure or different functions, they're actually mostly achieved by proteins. Okay, so this is how DNA can determine, everything in your body, right? How you look, how tall you are, what your hair color is, what your eye color is, how you function. Can you jump really high or can you run very fast? Okay. So again, ultimately, it's determined by what kind of proteins you have, but ultimately it goes back all the way to what kind of information is in your DNA. Okay, so the RNA can perform all kinds of functions, but the three main ones are here. So we have a messenger RNA, mRNA. So they are going to transfer or work as a messenger to bring the information from DNA out of the nucleus into the cytoplasm where protein synthesis takes place. Okay, so they are like a little messenger, right? Working for DNA. Okay. Ribosome RNA. So the rRNA is a major component in the ribosome. Okay, so the ribosome has ribosome RNA, which will perform protein synthesis. Okay. And then transfer RNA. So when you make proteins, these transfer RNAs, tRNAs, will bring on the amino acids. If you remember, that's the building block for proteins. They will bring the amino acids to ribosomes to, make the proteins. We also have a microRNA. There you go. So these mRNAs have, they're amazing. They can kind of regulate the gene expression process. So we're going to talk about that in more details when we get to that particular chapter. Right. Just more information about RNA. I already mentioned that, right? So they're the messengers for DNA. So what happens is this is your DNA. Remember, you have the double helix structure, so you have two strands. And then when you need to kind of transcribe information from DNA to RNA, the two strands of DNA will separate, and then you're going to use that base pairing rule or principle to make RNA, mRNA, based on the gene sequence in DNA. Okay, so, and then for RNA, we're going to talk about, that's the main component in ribosomes, right? The machine that produces proteins. So sometimes it looks like this. You have two subunits, a small one and a bigger one. And these RNAs can work with the proteins some enzymes, right? Most enzymes are proteins to make new proteins. Right? And then tRNA, that's the basic shape of tRNA. It almost like a clover leaf. Up amino acids. See over here, that's an amino acid attachment site. So it will kind of go around, pick up the amino acid, and then bring the amino acids to ribosomes to form a long polypeptide chain. Okay? And then we talk about this, the microRNAs, they're involved in the regulation of gene expression. Okay, so we'll talk about in the gene expression chapter. So here's a quick comparison between DNA and RNA. Let me just point out a few things. Okay, now this is important. Pyrimidines, so instead of T, you have U in RNA. So remember that this is a major difference. And sugar, also different, right? Deoxyribose in DNA and ribose in RNA. Structure, DNA is double-stranded. Okay, remember that there are two strands in DNA, but RNA molecules are usually single-stranded, just one strand. So a lot of times the DNA is more stable than RNA because you have two strands. Location, DNA is located inside the nucleus. It never comes out of the nucleus. Okay? So that's why you need messenger RNA to kind of bring the message out of the nucleus to the cytoplasm where protein molecules are produced. Okay. But mRNA, once it's synthesized, mRNA, it can exit the nucleus and go into the cytoplasm to deliver the information. Okay. All right, and then function, we already talked about them. So the central dogma. So you know what? I'm gonna save this for the DNA and the gene expression chapter. So let's skip this slide and then we'll go back here so you guys can read that. I think mostly just what the macromolecule is, what the monomers or subunits are for that particular micromolecule. Remember some of the examples, and then the functions for the particular micromolecules.